Good morning, everyone. Welcome to the lecture in the series of molecular biology. Today we'll be talking about the mutations and the mutagens. And see, the complete lecture that we are talking about in terms of mutations and mutagens, what all we'll be talking about in this scenario. See, the main objective of this lecture is to tell you what are mutations, what are the types of mutations, what are the different chemicals that can cause these mutations that we call them as mutagens, and what is the molecular mechanism, what are the different molecular mechanisms that are attributed in onset of mutations. So by the end of the lecture you will be able to understand the role of all these scenarios. So what is mutation? See, people describe them in different terms. Certain heritable change, certain inheritable change is called as mutation. What I define is that a permanent change in the sequence, a permanent change in the DNA sequence, that is mutation. See, what you are describing, certain inheritable change. The change is happening in DNA sequence. So you have to clearly define that yes, a permanent change, not a transient change. A transient change is not termed under the category of mutation. A permanent change which has altered the nucleotide sequence of the DNA is called as mutation. Now, depending upon the cell type and depending on the different types of variations, there are different categories of mutations that we're talking about. See, mutation in our system, we have two different types of cells, broad category, the autosomes and the sex chromosomes, like the gamete cell and the somatic cells. So mutations can happen in both the type of cells. Mutations happening in somatic type, called we are calling it as somatic mutations. Now, see why mutations, why we are talking about mutations and what are the consequences of mutations. See, we while studying the gene and genome component in the complexity, we had a clear-cut understanding that 97% is non-coding and 3% is coding. Suppose if any change or permanent change is happening in the coding region in such a manner that it is distorting the expression, that means that the phenotypic appearance of that mutation will be seen. Why? Because it's in the coding gene, and coding gene is responsible for the secretion of protein or for the expression of protein, and that protein is involved in some function inside the cell. So if the gene is now mutated, it is not able to form a proper protein, and the function is impaired. So you can clearly see that this is the impaired function. But if the mutation is happening in the non-coding part, and among the non-coding part, if it is happening in a region which is not involved in any kind of regulation or any kind of enhancing activities, it is happening in the repetitive part, then there will be no effect. Why? Because they are coding for nothing as of now, right? So those mutations which are happening in that region are called as neutral mutations because they are not giving any kind of phenotypic appearance, right? So what are the agents that we'll be talking about? See, agents can be physical or it can be chemical. There are multiple chemical agents that are termed as mutagens. Like there are certain intercalating dyes, there are certain chemicals that can directly modify the DNA molecule. And then there are radiations that we're talking about, whether it is ultraviolet or whether we are talking about the gamma radiation exposure or the beta radiation exposure. Those all radiations have been correlated in correlated with the mutation, correlated with the alteration in the DNA sequence, correlated or directly related to the damage to the DNA, damage to the DNA. So that is one of the causes of the mutation. What can be the other causes? Other causes, see, the most prominent mutation in our body is spontaneous mutation. The mutation which is happening because of the replication enzyme, that is polymerase. While we were reading the DNA polymerase or the DNA replication part, we clearly established that there is a chance of error, and that error is repaired by the repair pathways. But if somehow the mutation happened in the enzymes which are responsible for the repairing of that error, then what will happen? That error will be static in the DNA sequence and it will be propagated. It will be propagated. No repair has happened. That may also lead to mutation. So when you're talking about somatic mutation, then there are because of the mutation there are multiple pathogenesis of cancer that has been correlated. And when you're talking about the skin cancer or the blood cancer in form of leukemia, it has been shown because of the result of somatic mutations. So it's not that the mutations are always harmful. See, when if we talk about the evolution part, see, evolution means survival of the fittest in Darwin's term. What made us adapt to the harsh condition? If an organism is there, if organism is persistently exposed to high saline condition, that eventually after multiple generations, it 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 starts growing in that particular harsh condition. So what is there that has altered that particular behavior? That is that there is an adaptation. There are mutations that happen in DNA which are responsible for the tolerance. That means those mutations were in fact helpful for an individual to survive. Helpful for individual to survive under certain conditions. And that's how evolution progress. That's why we say mutation is the driving force of evolution, and that is the reason that we categorize mutations in either positive or negative, although beneficial or harmful. Although majority of the cases we talk about is harmful, but the beneficial mutations may also happen, which can give a certain survival advantage over the existing individuals of the same species. So although that case scenario is very rare, but that again that can happen. Then, what are the classes of mutation that we're talking about? There are spontaneous mutations which are caused because of the replication in the DNA, because of the error incorporated by the DNA polymerase, or because of the deficiency that the error is not being repaired. And the frequency of spontaneous mutation is very low. That's why you see sometimes that even a healthy individual which has no addiction problem, which is not never exposed to any kind of mutation, develop cancer. And why the cancer is developed? Because of spontaneous mutation. The mutation can happen in any gene, in any cell type, that may lead to aggravation and pathogenesis of cancer. If it's happening in the if it happens in, suppose, tumor suppressor gene or P53, the P53 got mutated, and the cell type in which the P53 got mutated is now growing as a tumor. Or it may be induced mutations. Induced means there is an external stimulus which has brought about the change in the DNA sequence. Can be UV light, can be any chemical mutation, can be any kind of other radiations like X-rays or gamma rays. So when we'll be talking about the molecular mechanism, we'll be talking about the mechanistic part also, that how these radiations are causing the mutation. But for now, let us talk about types of mutation. And for types, I strictly categorize them into two different parts: chromosomal mutations, genetic mutations. See, genetic, although genes tightly woven in a nucleosome model to form a chromosome, but then again, if we are talking about the structural integrity part, chromosome is a separate structural integrity part, what I'm talking about, and going inside the chromosome and altering the sequence of the DNA by manipulating the genes or by manipulating the nucleotides, those mutations are separate. So first we'll be talking about the chromosome mutation. In chromosome mutation, there are 5 types: deletion of a small segment of a chromosome; inversion, the deletion is now inverted; or translocation, non-disjunction, or duplication. What are these? Let us talk. First, we'll talk about deletion. See, before going to this, the most important mutations that one should understand are indels, INDELS, insertions and deletions. It's because of the insertions and deletions the evolution is progressing ahead, and you see different variations. So when we're talking about deletion, that is that from a chromosome, a small segment is excised, is taken apart, and then this particular chromosomal segment is now deleted. Its genetic material is deleted. So you see the length of the chromosome is also shortened. The length of the chromosome is now shortened. So a segment of DNA is gone. The segment of DNA which was tightly bound as a chromosome is now lost. If in this segment there are multiple genes that are actually responsible for certain functions, now the mutated organism will not be having that type of function. So deletion leads to removal of genes that is causing removal of the function or impairment. Then we are talking about inversion. Now what is happening in inversion? A chromosome segment is breaking from a fragment is breaking from a chromosome, and then it is performing a flip movement thereby inverting the sequence and then again reinstate it. So when the sequence is in a certain direction, now the sequence is breaked off, it is getting inverted and then again realigning. So the complete reading frame is now inverted. It was supposed to form a straightforward protein. Now, since the sequence is inverted, then the gene is now become will not be able to form a potent protein. It may form a truncated protein. So this is called as inversion, and these type of mutations are seen in the chromosome scenario. Then duplication, and this is very common. This is very common. Duplication of gene. You will see that a segment of gene is present on chromosome 1 also, and it is present on chromosome 6 also. How come a gene is present in two copies on different chromosomes, or it is present in two copies in the same chromosome? That is called as duplication. That is called as duplication. What is duplication? See, initially the chromosome is there. Then what is happening? The same segment is now duplicated and attached to the adjacent site. This is called as the DNA duplication part. This is called as the DNA duplication part. Then next is your translocation. Then next is your translocation. Now what is translocation? Transferring a segment from one chromosome to other. Transferring a segment from one chromosome to other. See, these are two chromosomes. Now what is happening? There is an exchange of fragment. There is an exchange of fragment between the two chromosomes. So you see a small portion is attained and a small fragment is given. Thereby you see that there are there are segments exchanged from the chromosome. That is called as translocation. Now please take a note in this slide. In this referral, when we are talking about normal translocation, we are not talking about the translocation of the centromere or the kinetochore or centrosome that we are talking about. It's the fragment which is present either in the long or the short arm that is getting translocated, right? So when we are talking about another set that is called as non-base junction. Now what is non-base junction? For this you should first remember what is happening in metaphase, after metaphase, anaphase, what is happening in anaphase, or what is happening in anaphase 1 of meiosis. The spindle fibers are attaching to the kinetochore or the centrosome, and then they pull the chromatids apart pull the chromatids apart in meiosis 2. That is what happening here, right? The spindle fibers responsibility is to pull the chromatids apart so that equal distribution of chromosome can happen in the daughter cells. Now suppose the while pulling the chromatids were not able to get separated and they are still attached to the centrosome or the kinetochore. They are still attached to the centrosome or the kinetochore in such a manner that the complete separation has not happened. The complete separation has not happened. So a proper disjunction that was supposed to happen is not happening, and then the complete two molecules of a same chromosome are now entering inside a single cell. So what will happen? It will either increase a copy number from one type, causing the trisomy part or ploidy level, and then it might go for nullisomy and it can remove the chromosome. It can show a removal of chromosome. So that happens in the meiosis part in terms of non-base junction. Then what is another case? So if you summarize, then we talked about deletion, we talked about inversion, we talked about duplication, and then we talked about the translocation part and non-base junction part. So these 5 things we have collectively termed right now under the chromosomal mutations that we should talk about. Now, let us focus on this particular slide at how deletion is going about. When we are talking about the genetic mutation, see, in the chromosomal part, I never talked about any kind of sequence etiology. Now, what that particular change in the fragment of DNA can cause? What can happen if this particular duplication is there? What can happen if the exchange is happening? So that is the scenario what we are trying to represent here. See, deletion will lead to the loss of gene. Then inversion, you see, Now it is reverted, the protein sequence is reverted, it will not be able to form. Translocation, it might be the case that yes, a segment can be translocated to other chromosome, and then it can lose the integrity, or it can lose the complete sequence. Then the last was that is shown in this particular animation is duplication, where a new copy is inserted. Now, with the new copy inserted, it might gain multiple copies. You might gain multiple copies, and you can say that yes, multiple proteins will be there. But if the intergenic sequence is duplicated, then you are losing the function. Then you are losing the function. So that is what we are talking about in terms of your chromosomal aberrations. Now, apart from these chromosomal aberrations, there are multiple mutations that we'll be talking about. And before going to the genetic level, you should have a clear-cut understanding of the mutagens. What are mutagens actually? What are the components that are inducing mutations? So, see, uh, when we talked about the type of mutation, I said there are two types, induced and spontaneous. For spontaneous, we are not talking about mutagen because it is being acted, it is being conducted spontaneously. There is no driving force, it's an error. Error is not deliberate. When we are talking about mutagen, that is induced. We are talking about induced mutations, and the components or the compounds, chemicals, the molecules that can alter the DNA sequence, that can alter the nucleotide sequence of a DNA. I had said alter. I never said deletion in deletion. I said alteration. Alter the nucleotide sequence of the DNA. That is called as mutagens. So that mutagens can be biological mutagen, chemical mutagen, physical mutagen. See, it can be radiations part with physical. It can be chemicals, and then there are certain agents that can cause mutations, biological agents that can cause mutations, like certain viruses, classical hepatocarcinoma, the persistent presence of hepatitis virus in the liver that can lead to hepatocarcinoma, and people have seen that 50%, 15% of the cancer types of hepatocarcinoma have been attributed to the presence of hepatitis virus. And not even hepatocarcinoma, multiple type of carcinomas have shown the history of viral infection. And that viral infection, because of its ability to integrate in the genome or alter the genome, they are giving rise to that particular mutation. And that's why we separately study the oncogenic viruses. That's why we separately study the oncogenic viruses, where they are modulating the tumor suppressor genes. And those oncogenic viruses, whether it is DNA oncogenic virus or RNA oncogenic virus, we classify them in terms of biological mutagens. So that is what happening. That is what happening. And the principal thing that you should be able to understand, whether it is a biological agent, whether it is a chemical agent, whether it is a physical agent, they need to alter the DNA sequence. For altering the DNA sequence, first of all, they should have the ability to cross the body barrier, enter the cell, enter inside the cell, and then reach the nucleus, and in the nucleus target the DNA. So they should be able to cross the membrane barriers. Now the radiations, which are highly ionizing radiations, only they have that much linear energy transfer ability, so that they can cross the barrier and can deposit the energy directly to the DNA molecule. Whereas the chemicals, if they are to cause mutation, they should be able to cross the membrane barrier and reach the DNA. And similar is the case of biological agents. Similar is the case of biological agents. So, what are the types of mutation? Let us go in brief. We are talking about the physical mutation, like radiations, like UV rays, X-rays, gamma rays, or we are talking about the chemical mutagens. Chemical mutagens like base analogs, what we are talking about, 5-fluorosyl, 5-fluorouracil, or alkylating agents like dimethyl sulfonate, or intercalating agents like ETBR. So all these agents are responsible for altering the sequences. Base analogs. Now these base analogs will mimic a natural base, that is ATGC. They can mimic any of the base, and then they can go and incorporate themselves inside the DNA while replication, and thereby they can cause a mutation or aberrant expression. Or alkylating agent, they can alkylate the base which is present. They can alkylate the cytosine residue. Thereby, they can the repair pathway will recognize that and then add or incorporate the thymine in place of it. Then intercalating that, they can go and attach in between the DNA sequence, thereby causing the mutation part. So, see, some products with possible mutation, I just incorporated a slide, nothing offensive against the brand, but then again, if you remember that past few years, I guess 6 to 7 years back, it was reported that there is a high content of lead in noodles or Maggi noodles, which can lead to the certain form of carcinoma. And then there was a scrutiny on that particular brand, and the complete products were wiped off, were taken back. And then again they went for the politicians and said that yes, lead is not there in the lead is not to the alarming level in that reported MAC part. Then certain air fresheners. Now these air fresheners are made up of chemicals like dichloropenzene or dichloropropanol. These chemicals that are used in these air fresheners, they are very potent mutagens, and they can cause the mutations of the bronchial cells of the lung. Or we are talking about the nail polishes. They contain dibutyl phthalate, which is a known skin cancer agent or a basal cell carcinoma agent. So you see, there are multiple mutagens that are present here. I just have discussed a few classical brief about the things, and then when we are talking about how these mutagens are actually causing mutation, let us discuss one by one. Let us discuss one by one. See, the first of all, let's talk about the UV light or the UV damage. Simple question. The most simple question for the people who are regularly coming into the lab. Why you switch on the UV before working in laminar or any kind of biosafety cabinet? Why? Simply because it is being narrated to you that if you switch on the UV, it goes for the sterilization process. It kills the contaminants. How? How it kills the contaminants? See, the exposure to the UV, the exposure to the UV radiation, what it is doing, it causes the formation of pyrimidine dimers. It causes the formation of pyrimidine dimers. What pyrimidine? Specifically, I am talking about the thymidine dimers. It will cause the dimerization of thymine. So the dimer, both the thymine residues will be forming a dimer, and since they have formed the dimer, there will be a distortion. There will be a distortion in the backbone of the double helix DNA, and this distortion is actually recognized by the repair pathway, and then they come and cause the repair. If the repair is not there, then this thymine thymer is removed and the mutation is attained. So what they do, it results in the formation of a cyclobutyl ring. It results in the formation of a cyclobutyl ring, and these dimers actually cause this particular harm that is called as buckling activity, cause formation of a nick. Now, if these dimers are able to replicate, if they replicate, then they become very lethal part, and then they eventually lead to the death of the cell, or if the damage is not able to possible. This is how the dimerization is happening. So if the dimerization has happened, these two corresponding adenine residues are also free. So there is a clear cut nick or the distortion in the backbone that is visible. Then when we are talking about the chemical mutagens, see, chemical mutagens, there are three different mechanisms what we are talking. Either you will be going for substitution, that you have taken the base out, you have removed the cytosine, and you have added the thymine. That is called as base substitution. Then either you are modifying the base, or you are actually causing the damage in the DNA, And based on this only, there are different repair pathways that are categorized, right? So let's talk about the replacement. What is happening? Taking an example, we are talking about the base analogs here. So we have four bases ATGC. Consider a compound called as 5-bromouracil. Now our DNA polymerase is not able to distinguish between the actual ATGC and the base analogs. That's the concept. It will not be able to distinguish between the base analogs and the ATGC. Similar concept that we had studied in thymidine kinase when we were talking about the antiviral designing. So now what will be happening? The DNA polymerase is not able to distinguish then this particular molecule, the base analog, will be incorporated in the newly synthesized DNA, and these particular molecules can now cause either the transition or any kind of transversion, what we're talking about, or they can cause any kind of alteration in the sequence. So few classical examples I've noted here, like 2-aminopurines and 5-bromuracil. The concept is they are structural mimic, they're analog, so that the DNA polymerase is not able to distinguish. So if we are talking about the 5-bromuracil, what it is doing, see, it's a complete analog of this particular of thymine. If you look at the structure, it's a structural analog or the base analog of thymine. So instead of a methyl group at the fifth position, instead of a methyl group, there is a bromine residue which is present on the 5' end. That is the only difference. So what it is happening, what will be happening? It will going and bind to it will form a bond, double bond with the adenine residue. So instead of AT, now there is a bond of A and 5-bromuracil. 5-bromuracil. Although in certain rare cases, it can also pair up with the guanine residue. It can also pair up with the guanine residue if it's in the ionized form. So if there is a 5-bromouracil which is formed, so repair pathway will come and try to remove it, then the situations will be normal. But if the repair pathway is not able to form a normal situation, then what will be happening? Then what will be happening? Then this particular strand which is formed, that is 5-bromuracil, will be recognized as a mutant part, and it will be propagated as a mutant. It might be propagated as a mutant. So that is the mechanism by which this particular 5-bromuracil is got into effect. Then let's talk about 2-aminopurine. Now 2-aminopurine, if we are talking about, see the structure of 2-aminopurine, it's this analog of adenine. It's the analog of adenine, the 2-aminopurine. Now, instead of adenine pairing with the thymine, this particular analog can pair with the thymine part, and thus it will cause a transition mutation. Mutation, and also it can mismatch repair. It can also mismatch or bind to the C residue also. Next is few of the agents like alkylating agents and hydroxylating agents or deamidating agent which can modify the base. How? We are talking about the base here. We are not talking about the nucleotide. What they are doing, they are modifying the adenine, the guanine, the thymine, and cytosine, which are attached at the with the deoxyribose sugar. So how the base can get modified? If you add a methyl group or any alkyl group, that is called as alkylating agent. Or if you are adding a hydroxyl residue OH, that is called as hydroxylating agent. Or you are removing the amino group, that is called as deaminating agent. So all these agents can modify the bases, and thereby they can induce the mutation. Like classical example of EMS, very potent mutagen regularly used in lab also. That is ethyl methane sulfonate. Now what it is doing? It will add an ethyl group to the guanine residue. It will add a ethyl group on a guanine residue, and it will make it in the form of ethyl guanine. Now, since G was supposed to pair with C, if it's methylated guanine, then it pairs up with T. So you see the position where GC was to come, now that instead of C, T has come. Instead of C, T has come. Now the repair pathway will come. It will see, oh, there is a mismatch. There is a mismatch, and the repair pathway will see that T is normal and modified residue we have to remove and add A. So instead of GC, now TA will come. So that's the transition mutation that is caused. That's the transition mutation which is caused. And then if it ethylates the thymine, then the transition mutation changes from TA to GC, and this is how they are causing the mutation. It will add the ethyl group, so there will be a bond between the guanine, ethyl guanine, thymine. The bond should have been between the ethyl guanine and cytosine, but now it is with thymine. So eventually repair pathway will come. It will see that this is normal, this is abnormal. Let's remove this part and add a fresh adenine. So what has happened? Initially, which was supposed to be GC, now has been converted into AT. Or Vice versa, if it's going on thymine, then there are hydroxylating agents, that is hydroxylamine. Hydroxylamine will actually add the hydroxyl group. It adds the hydroxyl group to the cytosine residue. It can add to both actually. It can add to cytosine also, it can add to guanine also. Let's talk about cytosine. If it adds with the cytosine, instead of pairing with G, it pairs with A. It pairs with A. So the transition mutation is happening from CG to TA. Now, if it's CG to TA, and suppose it is happening at the first position of the codon, then it will alter the amino acid. It will completely alter the amino acid. And if it is happening at the third position, it might be the case that because of the wobbling nature, it might code for the similar amino acid, or it might also code for a different amino acid. So that is the case that is happening. And then when it is hydroxylating, this particular guanine, or whether we are talking about alkylating agent in this particular guanine using MMS also, then also it causes the transition from GC to AT. So next in the category, or perhaps the last in this category, is nitrous acid, that is very potent chemical of mutagen, and sodium bisulfite. Now, why I have choose this? Because it's a classical example of deamination. See this cytosine residue. If you remove this NH2 group and add an oxygen to a keto form, it is converted into uracil. So what is happening? There is a deamination reaction. So instead of cytosine, now it will be uracil that is incorporated. Now it is the uracil which is compound. Now please remember, cytosine pairs with G, uracil pairs with A. So there is again a transition part. Or if you remove the amino group and the methyl group from 5-methyl cytosine, it gets converted into thymine. So the 5-methyl cytosine, if there is a deamination, it converts into thymine. So if a simultaneous reaction of methylation as well as the deamination is performed, then you will see the cytosine is converted into thymine and the mutation is induced. This is how the complete change is brought about by the nitrous acid. Now, nitrous acid is actually induced oxidative based deamination. Now, what is oxidative based deamination? It's a very potent oxidant that causes the oxidation of the substrate. So, what will happen? It will act on adenine nucleotide or the base. Then it will cause the oxidation to form hypoxanthine, and that hypoxanthine can pair with C, that is cytosine. Instead of T, it will now pair with C. Then it can also cause deamination of cytosine to form uracil that can pair with A. It can cause oxidation of again the pyrimidine residue, that is guanine, to give rise to xanthine. So from adenine it is hypoxanthine, from guanine it is xanthine. So always remember the procedure or the modification part, what they are applying here. So that is what we wanted to discuss here. And based on this discussion itself, you are now able to attain the set that what are mutations, what are chromosomal aberrations, what are the mutagens that we are talking about, what is the molecular mechanism that we are talking about. So that is all in this lecture. I hope you have now attained a basic understanding of the mutation part, and uh, you will be getting exposed to these terms again and again while studying the biotechnology or biology and biology. So please have a clear-cut understanding of this topic. So thank you so much. Thank you for your patience listening. I hope the lecture was fruitful to you. We'll again meet with another set of lecture in the same series. Till then, take care and wish you a happy learning.